Team number 17, DNA viruses. Plan of the team: the structure of DNA viruses, classification of viruses and properties of herpes virus and adenovirus, epidemiology of herpes infection, Epstein-Barr virus and cytomegalovirus infection, and the pathogenesis of herpes infection, Epstein-Barr virus and cytomegalovirus infection and adenovirus infection, and the clinical manifestation of herpes infection, Epstein-Barr virus and cytomegalovirus. Laboratory diagnosis: herpes virus. Herpes can appear in various parts of body, most commonly on mouse. Herpes simplex viruses belong to alpha subfamily of Herpes viridae. They are extremely widespread and exhibit a broad host range, can infect many types of cells and different animals. However, the human herpes viruses infect men. They replicate fast, 18-hour cycle, spread fast, and are cytolytic. And they can cause a spectrum of disease involving skin, mucosa, and various organs. And they undergo latency in nerve cells, reactivate later, causing recurrent lesions. And herpes simplex virus are of two distinct types: herpes simplex virus 1 and herpes simplex virus 2. They differ from each other in many aspects. Classification: human pathogens: alpha herpes viridae types: herpes simplex virus type 1 and herpes simplex virus type 2, and varicella zoster virus, and beta herpes viridae types cytomegalovirus and human herpes virus type 6 and human herpes virus type 7, and gamma herpes viridae Epstein-Barr virus. Herpes simplex virus structure. Herpes simplex virus: first, oropharyngeal contact with infected saliva, and herpes simplex virus 2, sexual contact or vertical mode from mother to fetus, and site of infection: herpes simplex virus replicates at the local site of infection and produces lesions anywhere, but more commonly in herpes virus. Herpes simplex virus first lesions are confined to areas above the waist, most common site around mouth, and herpes simplex virus 2 produces lesions below the waist, most common site genital area. Spread via nerve, virus then invades the local nerve endings and is transported by retrograde anoxal flow to dorsal root ganglia, and varies replicate further and then undergoes latency. Herpes simplex virus first structure: envelope proteins on surface and tegument and nucleocapsid inside of virus, and DNA and lipid envelope present on surface. And properties: two distinct herpes simplex virus type 1 and type 2. Their genomes are similar in organization and exhibit substantial sequence homology, and however they can be distinguished by sequence analysis or by restriction enzyme analysis of viral DNA. The two viruses cross-react serology, but some unique proteins exist to each for each type. And the pathogenesis of this disease: this is latent infection. Herpes simplex virus has a tendency to undergo latency in neurons, and herpes simplex virus first undergoes latency in tegmental ganglia, and herpes simplex virus 2 undergoes latency in sacral ganglia. And the non-replicating state. Herpes simplex virus doesn't replicate in latent stage except for a small RNA called microRNA, which maintains the latent infection and prevents cell death. The virus cannot be isolated during latency. And entry by skin or mucous membrane. Next, viral multiplication, local viral multiplication, and next lysis of cells and sensory nerve. They enter to sensory nerve and the root ganglia, and next latency. And lysis of cells appears after lysis of cells appears vesicles, and after vesicles ulcers, and reactivation develops. Herpes simplex virus type 1, classic presentation of primary herpes simplex virus 1 is herpes gingivostomatitis, oral mucosa. Herpes simplex virus first primary infection, usually mild. 10% of cases can be severe, and in early childhood. And herpes simplex virus direct contact with mucosa or abraded skin, and latency in trigeminal ganglia, and age-affected young children. Antibody distribution present 90% of people, especially adult, and the common manifestation oral facial mucosal lesions, and encephalitis and meningitis, ocular lesions, and skin lesions above the waist. And neurovirulence less, drug resistance less, and egg forms smaller pox. Herpes simplex virus. This slide shows herpetic aphthostomatitis in children. And herpes simplex type 2. More than 3.7 billion people under the age of 50 or 67% of the population are infected with herpes simplex virus type 1. And according to World Health Organization, herpes simplex virus is characterized into two types: herpes simplex virus first and second type, and both herpes simplex virus 2 are highly infectious and incurable, and herpes simplex virus 2 is almost entirely sexually transmitted through skin-to-skin contact and causing genital herpes. And the herpes simplex virus 2 sexual contact or vertical mode from mother to fetus. In the epidemiological patterns of herpes simplex virus 2, herpes simplex virus 2 is transmitted by sexual or vertical routes, and primary infection occurs in adult life. Antibodies develop only in 20% of people, particularly among black women than men and whites. And herpes simplex virus 2 tends to recur more often than herpes simplex 1, irrespective of the site of infection. And genital lesions. Herpes simplex virus 2 is more common than herpes simplex virus 1 to cause primary as well as recurrent genital lesions. Genital lesions are described as bilateral, painful, multiple tiny vesicular ulcers, and this may be associated with fever and lymphadenopathy. Primary genital infection with herpes simplex 1 and 2 are usually asymptomatic, and the classical clinical features consist of macular of papular skin and mucous membrane lesions occurring approx. 7 days after sexual contact, these progress to vesical pustulus and the ulcers and can last for up to 3 weeks. And typical symptoms also include pain, especially painful inflammatory swelling of the vulva in women, and the burning pain and the dysuria. And lymphadenopathy, fever, and cervicitis in women, prostatitis and in men, and are relatively common associated symptoms. Genital herpes may manifest atypically, particularly in the flame genital tract, making the clinical diagnosis form more difficult. Signs of herpes lesions of the cervix are relatively commonly in the absence of symptoms, while urethral manifestations are often associated with severe micturition problems. And varicella zoster virus produce vesicular eruptions, rashes on the skin and mucous membrane in the form of two clinical enteritis. And the chickenpox, it is characterized by generalized diffuse bilateral vesicle rashes which occur following primary infection, usually affecting children. Chickenpox, first step, inhalation of aerosolized virus, and the next respiratory mucous membrane damage, and the T-cell rich draining lymph nodes, and the first phase of replication, and the PBMCS, the primary T-cell associated viremia, and the reticulo and the endothelial cells in the liver, spleen, and other organs, the secondary T-cell associated viremia, and the next process is skin appears varicella, and the last process is latency in the trigeminal ganglion and the dorsal root ganglia. And shingles, zoster or shingles. It occurs following reactivation of latent varicella zoster virus present in the trigeminal ganglia that occurs mainly in adult life. Vesicular rashes are unilateral and segmental, confined to the skin, innervated by a single sensory ganglion. The first sign of shingles can be tingling or painful feeling in an area of skin, and headache or feeling generally unwell. And zoster or shingles. Zoster usually occurs due to reactivation of latent varicella zoster virus in old age, 60 years old, in immunocompromised individuals, or occasionally in healthy adults. And it usually starts with severe pain in the area of skin or mucosa supplied by one or more groups of sensory nerves and ganglia. And rashes, they are unilateral and segmental, confined to the area of skin supplied by the affected nerves. Most common nerve involved is also ophthalmic branch and of trigeminal nerve. Head, neck, and the trunk are the most common affected sites. Complications of zoster. Post-herpetic neuralgia is the most common complication in elderly patients. Zoster ophthalmicus, unilateral painful crops of skin, rashes surrounding the area. And visceral disease, especially pneumonia, can occur, which claims for the most common cause of death in zoster patients. Oncogenic action of herpes viruses is Epstein-Barr virus. Adenoviruses cause acute respiratory disease, usually pneumonia, acute follicular conjunctivitis, epidemic carotid conjunctivitis, cytitis, and gastroenteritis in infants, pharyngitis, and pharyngeal conjunctival fever are common. The icosahedral capsid is made up 252 capsamers, 240 hexons, forming the faces and the 12 peptones at the vertices, and each peptone bears a slender fiber. The double-stranded liner DNA is associated with two major core proteins and carries a 55 kilodalton protein covalently attached to its 5 ends. More than 100 oncogenic types of adenoviruses have been identified that infect mammals and birds. 47 human adenovirus types are classified. 5 more candidate types of presently studied. Infection may be productive, abortive, or latent. In productive infections, a viral genome is transcribed in the nucleus. mRNA is translated in the cytoplasm, and virions self-assemble in the nucleus. In latent infections and in transformed and tumor cells, viral DNA is integrated into the host genome. Virus host DNA recombinants are also found in productive infections. Infection is usually transmitted in droplets of respiratory or ocular secretions. Persistent infection occurs in the tonsils. Some adenovirus types are oncogenic in newborn rodents and can transform cell, and a few transformed human cell lines exist. Human oncogenesis has not been found, but may nevertheless occur by a hit and run mechanism. This slide shows clinical manifestation of herpes virus. Clinical manifestation of herpes virus is lesion of skin and lesion of mucous membrane, and ocular lesions, and visceral and disseminated herpes, and neonatal herpes, and cutaneous lesions, nervous system, and encephalitis, meningitis develop. And both herpes simplex virus first and second type have been isolated from nearly all mucocutaneous sites and viscera. However, in general, oral facial infections are common with herpes simplex virus 1, whereas herpes simplex virus 2 frequently causes genital infections are and this is the incubation period ranges from 1 to 26 days, median 8 days. Oral facial mucosal lesions are the most common manifestations of herpes simplex virus. Most common affected site in buccal mucosa, and the most frequent primary lesions are gingivostomatitis and pharyngitis. Most frequent recurrent lesions is herpes painful vesicles near lips, and other lesions produced are ulcerative stomatitis and tonsillitis. Many cases are asymptomatic but can predispose to secondary bacterial infection. Epstein-Barr virus. The term viral tropism refers to which cell types that Epstein-Barr virus infects. Epstein-Barr virus can infect different cell types, including B cells and epithelial cells. The viral three-part glycoprotein complex of GHGL and GP42 mediate B cell membrane fusion, and also the two-part complex of GHGL mediate epithelial cell membrane fusion. And Epstein-Barr virus that are made in the B cells have low numbers of glycoprotein complex, and because these three-part complexes interact with a human leukocyte antigen class second, and the molecules present in B cells in the endoplasmic reticulum and are degraded. And in contrast, Epstein-Barr virus from epithelial cell are rich the three-part complexes because these cells don't normally contain human leukocyte antigen class second molecules. As a consequences, Epstein-Barr virus made from B cells are more infectious to epithelial cell, and Epstein-Barr virus made from epithelial cells are more infectious to B cells. Viruses lacking the GP42 portion are able to bind to human B cells but unable to infect. Epstein-Barr virus structure. Envelope proteins on surface, envelope proteins present, and genomic DNA and tegument and capsid present. And Epstein-Barr virus is a member of gamma subfamily of Herpes viridae that causes infectious mononucleosis and is also associated with several human tumors, including nasopharyngeal carcinoma and Burkitt's lymphoma, Hodgkin's disease, and B-cell lymphoma. And antigens of Epstein-Barr virus are divided into three classes: latent phase antigens and early antigens, late antigens. And latent phase antigens, they are synthesized during the period of latency. Epstein-Barr virus nuclear antigen, it has 6 subtypes, and the latent membrane protein, it has 2 subtypes. And early antigens, they are non-structural proteins which help in viral replication. Late antigens, there are the structural proteins that form viral capsid and envelope. Cytomegalovirus family Herpes viridae, subfamilies alpha herpes virinae, beta herpes virinae, and gamma herpes virinae. Cytomegalovirus is the largest virus in Herpes viridae family. It is so named because it causes massive enlargement of infected host cell. And properties of cytomegalovirus are similar to any other herpes virus described earlier with some minor differences. Cytomegalovirus belongs to beta subfamily. Its dsDNA is the largest among herpes viruses, which consists of 140 nucleoids, and host specificity and cytomegalovirus are strictly species specific. Human cytomegalovirus doesn't infect animals. Similarly, a number of animals cytomegaloviruses exist which don't infect humans. Cell type specificity in vivo, cytomegalovirus infect kidney and salivary glands where it undergoes latency. In vitro, cytomegalovirus replicates only in human fibroblasts cell and fibroblast cell line, and produces a characteristic cytopathic effect described as owl's eye appearance. Cell-to-cell spread, cytomegalovirus is almost always closely associated with the cell and spread primarily cell-to-cell, so that very little virus may be cell-free. This slide shows information about cytomegalovirus. Cell culture infected with cytomegalovirus visible are massive intranuclear inclusions and separate from the nucleus enveloped by a light zone. Virus isolation, cell culture we use, and detection of cytomegalovirus DNA by PCR. Virus isolation, cytomegalovirus can be isolated from throat washings and urine. Human fibroblasts are the most ideal cell lines specific for cytomegalovirus. Cytopathic effect after 2-3 weeks of incubation, the following cytopathic effect may be observed in the infected cell line. Typical cytomegalovirus inclusions are described and multinucleate giant cells are seen, and the enlargement of infected host cell and shell viral technique can be followed for early growth detection. It involves centrifugation to cells culture mixed with the specimen to enhance the cell contact and viral application, followed by detection of early cytomegalovirus antigen in the infected cell by direct fluorescence technique. Diagnostic methods of viral infections: viroscopy, detection of viruses in the test material. Virus formats such as ELISA and rapid tests are available for detecting serum antibodies. Immunoglobulin M antibodies are indicative of active infection but appear only after 4 weeks of primary infection. Flushing from the nasopharynx contents or epithelium of vesicles, feces, cerebrospinal fluid, postmortal sections of tissue, immunofluorescence reaction, microscopy of histological changes, cell or nuclear inclusions in infected cell. Viroscopy, virus indication. Virus indication. Cytopathology is virus isolation. Virus isolation in various cell lines can also produce herpes simplex virus like cytopathic effects such as diffuse rounding and ballooning in of infected cells, and we use viroscopy and chicken embryo we use, and the sensitivity of all this method diagnose herpes simplex virus infection depends on the type of specimen as well as the type of infection. This sensitivity is more for vesicular lesions and primary infections than for ulcerative lesions and recurrent infections. Characteristic of cytopathic effect: diffuse rounding and ballooning in the infected cells, viral antigen detection by neutralization test or immunofluorescence staining with specific antiserum, and the shell viral technique can be followed to decrease the detection time to 24 hours. Viral antigen detection by direct immunofluorescence is also a sensitive and a specific assay. It can differentiate herpes simplex virus 1 from herpes simplex virus 2. The laboratory diagnosis of viral infections based on 3 general approaches: direct detection of viral components and the isolation of viruses, and the next demonstration of significant increase in serum levels of antibodies. Methods of cultivation of virus. Generally, three methods are employed for the virus cultivation: inoculation of virus into animals, inoculation of virus into embryonated eggs, and tissue culture.